Growth of bacteria. Under favorable condition, bacteria grow in size and number. Bacteria require nutrients and favorable physical and chemical conditions for growth. Phases of growth. When a culture medium is inoculated with a started culture and incubated, individual bacterial cell grow and divide by binary fission, increasing the population and size. The cell mass started increasing and doubles after every cell division. The pattern of growth can be studied by plotting time along x-axis and number of cells along y-axis. Typically, a bacterial growth curve has 4 phases: lag phase, log phase, stationary phase and death phase or autolytic phase. The first one is lag phase. After inoculation, individual bacterial cell require some time for adapting to the new environment and uptake for ingredients present in the medium. This is the establishment phase or lag phase of the culture, during which there is no appreciable increase in the population size, but the cells are not metabolically inert. They are actively synthesizing material for the upcoming division cycle. This is the lag phase. And the next one is log phase. Log phase or logarithmic phase is the period of active multiplication of growth. After the establishment phase, the cell mass start increasing and doubles after every cell division. The time taken for doubling is known as generation time. It varies from about 20 minutes to a few hours for most bacteria. As the population doubles by the end of each generation time, growth at this phase becomes exponential. Hence, the phase is also known as exponential phase. During the log phase, the generation time is extremely short and the cell divided rapidly. And the third phase is stationary phase. After the logarithmic phase, the growth rate falls due to the limitation of one or more constituents in the medium, accumulation of toxic or unwanted chemicals, changes in pH. During this period, increase the cell mass is almost balanced by cell death, so that the population size remains more or less static. The fourth one is death or autolytic phase. This is the final stage of growth of bacteria. In a culture medium, the cell die faster than new cells are produced, resulting in the drastic decline in the population size. During this phase, the number of viable cells decline exponentially due to the depletion of nutrients and accumulation of inhibitory products. Methods of growth determination. The first one is measurement of growth. Bacterial growth can be determined by various methods based on measuring any of the common parameters of growth: the cell mass, cell numbers and cell activity. Cell mass. Next one is cell mass. The cell mass of a bacterial culture can be determined by taking the weight or measuring the turbidity of the medium. The weight is determined after separating the cells from the medium by filtrating or centrifugation. In the turbidic method, the optical density of the cell mass in a unit quantity of medium at the suitable wavelength is measured. The content of principal cell constituents like DNA, RNA, total protein, peptidoglycan, etc. are also used to express the biomass. The next one is cell number. Different techniques are used to determine the cell number in a bacterial culture. This include direct microscopic count, plate count method, membrane filter unit, Electronic enumeration. Direct microscopic count. The cell number can be determined accurately by counting the number of cells after transferring uniformly suspended medium to the standard counting chamber like a Neubauer's chamber. Populations of microorganisms from natural substrates with high concentration of cells can be enumerated, diluting the substrate to a required level by using serial dilution. The next one is plate count method. This is the method used to analyze the viable cells in a microbial sample by plating them to a suitable medium. Plating can be done by using the pour plate method or spread plate method. A small amount of appropriately diluted sample is added and incubated. All the live cells in the sample will be multiplied to produce colonies that can be counted directly, and the results are expressed as the number of colony forming units, CFU. Membrane filter count. In this method, the bacterial culture is filtered through membrane filter to trap bacteria, and the filter is placed on the special counting pad saturated with appropriate medium. After incubation, the number of colonies developed is counted to determine the bacterial population. And the last one is electronic enumeration. In electronic enumeration, bacterial suspension is passed through an electronic particle count. The counting is done as the cells pass through a teeny orifice between two compartments filled with electrically conducting solution. As cells pass through this orifice, a resistance is generated in the electrical circuit, which is recorded and counted. The last one is cell activity. Here, the biochemical activity of a culture is estimated and it is considered as an index of bacterial growth. For example, production of a metabolic end product like an organic acid is estimated and considered as an index of growth. The assumption of that the amount of the product estimated is proportional to the magnitude of the growth. That's all. Thank you.